All right, let's take a second to talk about point mutations. So in Unit 5, we talked about chromosomal mutations or large-scale mutations that involved chunks of chromosomes or whole chromosomes and therefore multiple genes. In this case, what we're talking about are one little tiny change, usually in one base, that is going to result in a change in that one gene. It's not going to affect other genes around it for the most part. And so the first I can statement is I can describe the potential results of mutations. Mutations, when you hear the word mutation or mutant, you probably have a negative connotation. You probably think, oh, that's bad. But a mutation is simply a way to increase genetic variation, and it's not always bad. For example, the first human, whoever that was, the first Homo sapien, had a certain color eye, let's say brown, because most likely it was brown. Anybody who doesn't have brown eyes, that's due to a mutation. My eyes are blue. That means I'm a mutant, technically. Am I upset about that? No, because I like my blue eyes. But what it means is that original person had the gene for brown eyes, but over time there were mutations in that gene, and their children, grandchildren, great-great-great-great-great-great-grandchildren, whatever, that gave us brown. It gave us blue and green and hazel and all the pretty eye colors to go along with brown. So, not that brown's not pretty, it is. But all of those would be mutations, okay? So when we think of mutations, we need to stop thinking of it as a negative thing and start just thinking of it as a change in the DNA. It's really how we got everything different. The first person had a certain skin color, a certain hair color, a certain height, a certain build. And the fact that everybody on Earth now doesn't look like that. We all have different hair colors, different skin colors, we're different heights, we have different body builds. That's all due to mutations, and that's not a bad thing because mutations give genetic variation. And if you'll remember, we talked about the benefit of genetic variation is it increases the survivability of a species. So mutations in that way are very beneficial, just like sexual reproduction is. It increases genetic variation and increases the chance that a species will survive. All right, all that to say, when a mutation happens, a mutation is simply a change in the DNA. And when that happens, it can either be positive, it can actually help the organism, okay? It could be negative, it could hurt the organism, or it could be neutral, meaning that it doesn't impact the organism at all. And what is it helping, hurting, or not impacting is whether or not that organism survives and reproduces, because biologically speaking, that is the goal. So if I'm a squirrel out living my best little squirrel life and eating all my little acorns, my job is to try to stay alive long enough to make a litter of squirrel pups that have my genes and then get them to grow up to have more pups. So not only do I want to have the pups, I want to keep them alive long enough for them to reproduce. And that way my genes stay on. So a mutation that's positive is something that's going to help me survive and reproduce, or reproduce, I should say. Negative is something that's going to hinder that. It's going to prevent me from either surviving or reproducing. And then a neutral is something that might help or hurt me in another way, but it doesn't help me or hurt me as far as survivability or reproduction goes. So that's how we measure the benefit of a mutation is, does it increase survival and does it increase reproductive success? And if it does, then it's considered positive. All right, so there are two types of point mutations, and a point mutation is a mutation that happens at one point, okay? Whereas a chromosomal or large-scale mutation is at a big chunk. And so when we say 1 point, what we mean is one base. Okay, so here's my DNA. This is the same one we looked at in the last video, if you were there for that. Here you go. So DNA transcribed it into RNA, translated it into proteins using this codon chart, or codon wheel, if you prefer the wheel. That wheel's kind of pretty, so I prefer that. So the two types are a base pair substitution and a frameshift. A base pair substitution is where I'm going to take one out and put a different one in. Okay, and so I have substituted one base for another. A frameshift, if you'll notice, I wrote these in threes because when the mRNA is read, it is read in threes. Okay, and so that's called the reading frame. And so when you have a deletion, let's say that you delete that U, now my reading frame, because the ribosome has to read in threes, it can't say, oh, well, you deleted a U, I'm just going to read the GU and move on. It has to read in threes. So now it's going to read GUA, AAG, CUU, GA, whatever. And so that's why it's a frame shift, because it shifts that reading frame. All right, so I can describe base pair substitutions as a type of point mutation. So again, a base pair substitution is when you remove one base, take it out, and replace it with something else. And it is a point mutation because it is impacting one point in the DNA and therefore only one gene. Now there are three different types of, and I'm going to try to do these in different colors and not be confusing, but there are three different types of base pair substitutions. Okay, the first one, let's say that we change this to let's do missense first, TTG. Okay, that's going to change this from AAA to AAC. And then when I look at my chart, AAC gives me asparagine. Okay, so that's a missense mutation. A missense mutation is where you have a base pair substitution that leads in a different amino acid being brought in. Notice none of the others were impacted, just that one. This is what sickle cell, this is how sickle cell happens. All right, let me get a different color. So that's a missense. The next is I can describe a nonsense. Okay, what if I'm trying to think of how I can. A nonsense would bring in an early stop codon. And so an early stop codon, let's say that we did this one. So now it's A, C, T. Okay? So now I'm going to have U, G, A, and that's going to be stop. And so instead of my protein being 4 amino acids long, it's now 1 amino acid long. So you can imagine if it's 4 and it does all these foldings, then that is going to be, let me label these, missense, nonsense. That's going to be a problem because it's structurally not going to be the same. It can't fold and do things if it's just 1 amino acid. Now, like I said, normally proteins are hundreds of amino acids, and it might change it from 512 amino acids to 502 amino acids, or it might change it from 512 amino acids to 35 amino acids. I mean, it can be it depends on where that stop codon falls. Does it fall really early or does it fall later? As to how much it truncates or cuts short that protein. All right, the third one, let me see what other, got a little purple pen here. The third one is what we would call a silent mutation. And so with a silent mutation, let's say that we do this one. So we have C, G, T, okay? That's going to convert this to G, C, A. And when I look at this, G, C, A is still alanine. So even though there's a mutation, there's a change in the DNA, it does not change my protein. And so we call that a silent mutation because we don't see the effect of that mutation. Okay, there is a change in the DNA, there is a mutation, but because it doesn't change the amino acid, it's not going to change the protein. And so sometimes, I would say probably all the time, most of the time, we don't even know that mutation exists because we don't see it played out in the protein. The last is I can describe frameshift mutations as a type of point mutation. And so looking at a frameshift, let's see. So let's say that we delete this T. I'm not going to do it too. This pen is not, I'm going to have to use a highlighter. All right, so we went from TAC, ACA, TTCGAACT. Because what's happened is I've deleted this. Now I'm having to shift my reading frame. So my RNA is going to be AUG, UGU, AAG, CUU, GA, ... whatever comes after that. Okay, this is still methionine. This is still cysteine. AAG is lysine. Okay, so technically no change yet, but now we've got CUU. So CUU is leucine. So you can see this is actually a silent mutation, but now everything downstream, because we're not going to have a stop. GA is either going to be glutamine or asparagine. It's not going to be a stop. So our protein should be stopping here, and it's not going to. It's just going to keep going. And so you can see that's going to make a big difference in this protein. It's basically going to keep going until it runs into an accidental stop codon. And so that's a frame shift. And frame shifts are usually worse because Whereas a missense mutation, well, I shouldn't say they're always worse. With a missense mutation, you're affecting one amino acid. That may or may not affect the way the protein folds. Okay. Nonsense could be really bad because it might cut the protein really short. But a frame shift also can be really bad because it can mess up tons of amino acids. If you have a frame shift mutation early in a protein, you're going to have a bunch of problems. That protein's not going to be functional at all, or it's going to have a new function. Now, maybe that new function helps you, and then voila, we've got a mutation that's beneficial, but a lot of times it's not. A lot of times these mutations are the cause of a lot of genetic disorders like sickle cell and cystic fibrosis. And what are some of the other ones y'all studied? Maple syrup urine disease and progeria, and there's just a bunch of them. So I hope that helps with point mutations. If you've got any questions, though, let me know.